Hi everyone, my name is Zach Tom, and today I'm going to be presenting on bioinformatics metadata extraction for machine learning analysis. So let me start out with a little bit of background. Recently, research has shown that sequencing errors, also called artifacts, occur frequently in conventional next-generation sequencing workflows. And these artifacts are problematic because they appear to be relevant mutations, while in reality, they are artificially produced from steps of the NGS workflow and thus are not relevant. Consequently, artifacts negatively affect scientists' ability to detect relevant naturally occurring mutations. The important question then is which steps of the NGS workflow cause which artifacts to occur? To attempt to answer this, I used the NCBI Sequence Read Archive, or the SRA, as my source of sequencing data. And the steps of the NGS workflow that we're interested in are captured in the SRA metadata. Unfortunately, submissions to the SRA often do not contain the necessary metadata. This study here by Jay Onisser and A. Shanahan showed that less than approximately 20% of submissions contain keywords in any one of the three categories of fragmentation, adapter ligation, and enrichment, which are steps of the library construction protocol, which shows us that if we're trying to find this desired metadata out of approximately 450,000 submissions that exist in the SRA, doing so just by simply searching the database online would take way too long, which is why we need a tool that can do so efficiently, comprehensively, and programmatically, which I present to you as SRA Metadata X. So SRA Metadata X is a Python program that provides a command line interface for extracting metadata from the SRA. And it uses the SRA-DB SQLite database that's hosted by Bioconductor for its metadata. And this SRA-DB is continually updated with new submissions and new metadata. And so the development of this tool, SRA Metadata X, was the main objective of my project. However, I have also conducted experiments using it along with a variant calling pipeline and machine learning to demonstrate as a proof of concept what this tool can be used for. And here is an overview of my experiments. So first, researchers have extracted DNA from GM12878 cell lines and performed next-generation sequencing and then uploaded their data to the SRA. In the meantime, the Genome in a Bottle, or GAB consortium, has characterized the genome of this cell line, and this benchmark genome is called NA12878. Next, I used SRA Metadata X to search the SRA metadata for sequencing runs that have used NA12878 and have performed either PCR or hybrid capture for enrichment as a part of their library construction protocol. And then 8 of those extracted runs were fed to a variant calling pipeline that compares discovered variants with the high confidence variants identified by GAB in NA12878. These artifacts are finally fed to a machine learning model to elucidate a relationship between PCR/hybrid capture and the specific artifacts produced. So using SRA Metadata X, I searched the metadata for terms matching NA12878 and hybrid, and then NA12878 and PCR. And you can see here that this is like a snapshot of the results. And looking at, so now taking one of the results, ERR1831349, if we look at that page in the SRA, we see here that we do in fact have NA12878 being used, and they used hybrid selection for their target enrichment procedure. And so once these runs were identified, data was downloaded, and it was ran through the variant calling pipeline, and we finally produced our variants. And these variants come in the form of the chromosome number, the position on the chromosome, and the reference allele and the alternate allele. So I took that data, concatenated it into one data point, and then tagged on the binary column 0 for hybrid capture and a 1 for PCR. And that was the final form of the data ingested by the machine learning models. So in experiment 1, a logistic regression model was simply used as a first test, got an accuracy of 70% and AUC of 0.7. Then experiment 2 used a lot more models like KNN, SVM, random forest, multi-layer perceptron, did 10-fold cross-validation, got accuracies of 69 plus or minus 1%, and AUC of 0.77. So based on these results, the models were able to predict enrichment method decently well, indicating that these different enrichment methods that we've studied likely produce specific sequencing artifacts. However, the main objective of this project was to develop a tool that could extract rare metadata that other existing tools cannot. So the machine learning work was a proof of concept, and further experimentation with more data would likely result in higher accuracy and insight. Thank you.